Hey everyone, Dr. D here, and in this video we are going to be doing an Exam 3 review for my class. This is based on chapters 7, 8, and 9 from our Campbell's 12th edition biology textbook. So let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
All right, let's go ahead and get started with chapter 7, membrane structure and function. Now, just a reminder, please study all your slides. I just don't have time to go through every single concept. Obviously, go through the slides, learn the terms, learn the concepts, understand them. But what I'm doing here is just highlighting some of the more juicy concepts that are bound to be asked on the exam. Does that make sense? So don't just focus on this exam review. Please go through and learn all the handouts and all the concepts on those handouts to be, extra sure of doing well on the exam. So let's go ahead and get started with membrane structure and function. We started by talking about what a fluid mosaic model is. The membrane is fluid and it is a mosaic of different proteins that float in that membrane. And here you can see all those different proteins and lipids and glycolipids. Now, remember that membranes need to be fluid, and cholesterol serves as a fluidity buffer in the cell. That's important to remember. Oh, we need to know that cell surface membrane proteins, remember those transmembrane proteins? We need to know their jobs, their functions: transport, enzymatic activity, signal transduction, cell-to-cell recognition, intercellular joining, as well as attachment to the cytoskeleton, as well as the extracellular matrix. We need to remember that the membrane is selectively permeable. And you need to know what types of molecules can easily cross the membrane. Remember, you need to remember that it's those hydrophobic, non-polar molecules that easily or readily dissolve in the lipid bilayer and pass through the membrane rapidly to the other side. So you should know this includes hydrocarbons such as methane, CH4, carbon dioxide, oxygen. These molecules are small and nonpolar, and they easily cross across a semipermeable membrane. However, polar substances like sugars and water, they do not pass the membrane easily at all, and especially ions. Remember, cations, anions like sodium cation or chloride anion. These things do not cross the membrane. Okay, we need to know the difference between a channel protein and a carrier. Remember, channels are simply pores, while carriers, they change shape to accommodate the substance that's crossing the membrane. Remember that aquaporins are channel proteins for water. Remember the concept of diffusion, very important. The substances spread out evenly from areas of high concentration to low. Remember, a substance spreads from an area of high concentration to low until they reach equal concentration throughout the medium. If you have diffusion of solute, first of all, let's refresh our memory. Do you remember the terms solute and solvent? Solute are substances that are dissolved in solvent, right? So sugar is a solute, salt is a solute. Anything that's dissolved in water or dissolved in solvent is a solute. So here in this example, the dye, the molecules of orange dye are the solute, while the solvent is water. And do you recall these very important terms? If solute is diffusing across a semi-permeable membrane, like this dashed line in this example, then that is an example of dialysis. Remember, that's dialysis. If solvent, usually water, is what's crossing the semi-permeable membrane and not the solute, then that's osmosis. Do you recall? So which example is this on this slide here? Is this dialysis or osmosis? Right, Wicket. Hopefully you beat Wicket to the punch here. This is an example of dialysis because it's mainly these solute molecules diffusing across this semi-permeable membrane. So it's a diffusion of solute across a membrane. And remember, whatever is dissolving will dissolve from an area of high concentration of itself to an area of low concentration. So here, the orange dye is what's diffusing, I should say, diffusing, and the orange dye will move from an area of high concentration on the left to an area of low concentration to the right. There will be a net diffusion to the right until equilibrium is reached. Now remember that different solutes diffuse down their own respective diffusion gradients. So here, the orange solute is more concentrated on the left, so it will net diffuse to the right, while the purple solute, the purple dye, is more concentrated on the right, and it will net diffuse to the left until equilibrium is reached for both solutes. Remember the term passive transport? This simply means diffusion of a substance across a biological membrane. No energy is required. In fact, energy is released because passive transport is exergonic or negative delta G. Both dialysis, which is the diffusion of solute across a membrane, and osmosis, which is diffusion of water across a membrane, these are both examples of passive transport. And tonicity problems as well. Remember how cells react in an isotonic environment, hypotonic environment, hypertonic environment. All of these are examples of passive transport as well, though the tonicity problems are examples of osmosis passive transport. And again, osmosis is the diffusion of free water, so water molecules, across a selectively permeable membrane. Here, this example is, remember, this is an example of osmosis. Why? Because there is a selectively permeable membrane right here, but note that it does not allow the solute, which are these green molecules of sugar, they cannot cross the membrane. And when solute cannot cross the semi-permeable membrane, and only water can cross the semi-permeable membrane, this becomes an osmosis problem. And remember, osmosis, for osmosis problems, this is very important. Water will diffuse from where water is more concentrated to where water is less concentrated. Did you follow me? Water, whatever, remember, if anything, remember this that I'm about to say now. Whatever is diffusing, okay? Whatever is diffusing will diffuse from where it itself is high in concentration to where it itself is low in concentration. So take a look at this example here. Take a look. If the green dots could diffuse, okay, which they can't in this example, but let me ask you, if these sugar molecules, which are green, if they could dissolve, they would net flow to the left. Is that correct? The sugar would diffuse to the left. Why? Because the sugar is more concentrated on the right. And diffusion means whatever is diffusing will move from where it is most concentrated to where it is less concentrated, right? However, remember, the green dots cannot diffuse across the membrane. This is an osmosis problem. So now water is what's moving. This is what you need to understand. Water is what's moving. Water will do the same thing. Water will move from where water is high in concentration to where water is lower in concentration. On which side of this tube, on which side of this dashed line membrane here, is the water itself more concentrated? Here on the left where there's less solute, or here on the right where there's a lot of solute? That's right, Wicket. It's on the left. Obviously, if there's less sugar, that means the water is more pure. The water is more concentrated, right? So where the solute is lower, that's where the water is more concentrated. The water will diffuse from where water is highly concentrated to where water is lower in concentration. So there should be a net diffusion of water to the right. Okay, so hopefully this makes sense. If the green dots could have diffused across the membrane, that would have been a dialysis problem, and the green dots would have diffused to the left, where solute was high on the right? However, solvent's higher in concentration on the left, so water diffuses to the right. Please understand that. Here's another way of thinking about it. Whichever way solute wants to diffuse, the solvent wants to diffuse in the opposite direction. All right, remember, this leads us to tonicity problems, which are very important problems. This is the same concept of osmosis, but it's essentially how osmosis affects a cell. Remember, there are hypertonic environments, hypotonic environments, isotonic environments, and we need to know all of these. So let's just do a quick rundown here. In an isotonic environment, that means that the solution surrounding the cell has the same solute concentration as the inside of the cell. So in this case, for every one water molecule that enters the cell, about one water molecule exits the cell. And this is known, if it's an animal cell, animal cells prefer this, and this is known as a normal, healthy, happy cell, right? So remember, in isotonic environments, the solute concentration outside is matching inside, so there is no net flow of water because you're already at equilibrium, okay? And animal cells prefer this, whereas plant cells do not prefer isotonic environments. Plant cells actually become what's known as flaccid. So we need to know these terms: flaccid, normal, okay? And then in a let's go to hypertonic environment, the solute, remember solute, salt, the solute concentration outside of the cell is higher than the solute, or salt in this case, concentration inside of the cell. And remember, one more thing to remember is a Gizmo's tip for you, and that is that the solute is not what's crossing the membrane in these tonicity problems. Why? Because tonicity problems are osmosis problems. The water is what's flowing in these tonicity problems, not the salt, not the sugar, not the solute. The solute is not what's moving. The water is what's moving. And remember, water will move from where water is more concentrated to where water is less concentrated. And remember, inside of the cells, you're going to have more pure water than outside of this cell if it's a hypertonic environment. Hypertonic means high solute concentration environment. And if the surrounding solution is high in salt, well, water is going to diffuse from where water is high in concentration inside of the cell to where water is lower in concentration outside of the cell. So there will be a net efflux of water out of the cell, and this results in what's known as a shriveled or crenated animal cell. Whereas a plant cell, it's going to have a similar phenomenon, but it's a little bit different. Let me show you again. The salt concentration outside of the plant cell is higher than the salt concentration inside of the cell. So water will net diffuse out of the cell, and this causes the cell membrane to shrivel. Look at this line here. Look what I'm outlining with my mouse. The cell membrane shrivels and peels away from the cell wall. Look at this. The cell is dehydrating. The cell is losing water. But this part here, why does this part not shrivel up? What is this? Why does this not shrivel up? That's right, Wicket. It's the cell wall. It's too rigid to shrivel. The cell wall stays put, while the cell membrane is what peels away from the cell wall and shrivels up. This is known as a plasmolized cell. You need to remember that plants become plasmolized in hypertonic environments, while animal cells become shriveled or crenated in such environments, in hypertonic environments. Okay, conversely, look at a hypotonic environment here. In a hypotonic environment, the water concentration surrounding the cell is more concentrated. There's less solute surrounding the cell. There's less solute in the environment around the cell than inside of the cell. This means the water concentration outside of the cell is higher. The water will net flow into the cell. The cell will swell until it, that's right, pops and becomes what's called lysed. Lysed means broken. And in a plant cell, this is what's strange. Plant cells actually prefer it. Plant cells want water to rush into the cell. And so the plant cell can become nice and firm. This is known as a turgid or normal plant cells. This is the preferred state for a plant cell. All right, so now you know. And do you remember my trick to remembering these tonicity problems? The hypo sounds like hippo. Remember that? Hypo sounds like hippo, and that helps me to remember what? It helps me to remember that in a hypotonic environment, which is a low solute environment, in a hypotonic environment, my cells look like hippos, okay? Why would my cells look like hippos? Well, because water must be rushing in. Why would water rush in? Well, that means that water is diffusing in. And that must mean that the water concentration outside is higher than the water concentration inside, which means the solute concentration outside is lower than the solute concentration inside. All right, I hope this all helps you. This may take you a little bit of practice and writing it down. What I recommend is write it down and go through it step by step until it makes perfect sense. You can do it. So let's take a tiny little break time with Gizmo and Wicket so I can catch my breath here, and we'll carry on with more review time. All right, welcome back from break time with Gizmo and Wicket. Let's carry on with osmoregulation. Remember that the contractile vacuole allows cell wall-less organisms like paramecium to survive in hypotonic environments because it squeezes out any excess water that may have entered the cell. All right, moving on. Remember, when a transport protein needs to use energy to move solutes against the concentration gradient, this is known as active transport. So with passive transport, a substance is moving across a membrane down the concentration gradient from high concentration to low. In active transport, a substance is moving against the concentration gradient from low concentration to high. And this requires energy, usually in the form of ATP energy. And do you recall that the sodium-potassium pump is an example of an active transporter? Active transporters require energy in order to move substances from low concentration towards high. And then do you remember this summary slide here? These purple dots represent substances that can diffuse through simple diffusion across a semi-permeable membrane. And remember, for a substance to do that, it needs to be small and nonpolar, such as CO2, methane, and other hydrocarbons, oxygen, these types of small non-polar molecules. For ionic compounds and for polar substances, remember this requires a channel or a carrier protein. And when diffusion of a substance across a membrane requires a channel or carrier, this is known as facilitated diffusion. Facilitated diffusion, again, we need to know that for facilitated diffusion, we require a transport protein such as a channel or a carrier. For facilitated diffusion, this is still an example of passive transport because substances are diffusing across the membrane. And remember, these substances are either polar or charged, right? Okay, very important to understand those concepts. And then with active transport, remember, substances are moving against the concentration gradient, and this requires ATP or some other form of energy. Remember membrane potential, a voltage across a membrane. What if I asked you, what is responsible for producing a membrane potential? Remember, a membrane potential is formed when there's active transport of ions, such as a proton pump. You see here, this proton pump is pumping plus charges out. Well, if you're pumping plus charges out, you're going to get a voltage difference, more positive charge outside, more negative charge inside. So a membrane potential forms thanks to active transport of ions. Then with co-transporters, I just need you to remember that with co-transporters, this is when active transport of a solute indirectly drives transport of other solutes or substances. And then lastly, we learned about bulk transport. You should know that bulk transport includes exocytosis and endocytosis. With exocytosis, this is bulk export. With endocytosis, this is bulk import. And then I told you that phagocytosis is bringing in solids into the cell. Pinocytosis is bringing in liquids into the cell. And receptor-mediated endocytosis is when endocytosis is triggered by some sort of hormone or ligand that binds to a surface receptor on the cell. All right, and with that, let's move on to the next chapter, Chapter 8. All right, are we ready for Chapter 8, an introduction to metabolism? Let's go ahead and get started. Obviously, know the term metabolism, which includes all of an organism's chemical reactions, including the catabolic pathways, where energy is released when breaking down complex molecules, and anabolic pathways, where energy is consumed to build complex molecules. We then talked about different forms of energy. You should know all these forms of energy. What is kinetic energy? What is thermal energy? What is heat energy? And you should know that you should know that thermal energy is a form of kinetic energy. And then we talked about potential energy, which is energy based on a substance location or structure. And you should know that chemical energy is a form of potential energy based on structure. Next, we learned about thermodynamics, and you should know that organisms are open thermodynamic systems. Next, we learned about the first law of thermodynamics, that energy can be transferred or transformed, but not created nor destroyed. So you should know the first law of thermodynamics and what it states. Additionally, we need to know the second law of thermodynamics as well, and how it has to do with entropy. You should know that entropy, the units are capital S, and this refers to disorder. The higher the S value, the higher the entropy, the higher the disorder. The lower the entropy, the lower the S value, the lower the disorder. And you remember the second law of thermodynamics states that every energy transfer or transformation increases the entropy of the universe. So the universe is heading towards more disorder every time energy is transferred or transformed. And you should know why the bear, remember the bear is a representative of life here in this image. Why is life, which is so highly ordered, possible if the universe is said to be heading towards more disorder? Remember, the answer was that, well, the bear itself is ordered, but at a cost, because it produces disorder itself. When the bear consumes complex molecules in the form of sugars and fats, it then burns those complex molecules, releasing disorder in the form of heat, which is always disorder, as well as CO2 and water, which is disorder as well, because the CO2 and the water are byproducts of these complex sugars and fats. So the bear, which is a stand-in for life, does not negate the second law of thermodynamics, because it creates so much disorder, it pays the entropic penalty to the universe, allowing the bear to be ordered itself. Okay, next we learned about spontaneous processes. Remember, a spontaneous process has a negative delta G. It can occur quickly or slowly, depending on its activation energy. Remember, spontaneous processes release energy. They do not require energy input. Next, we talked about Gibbs free energy. Remember, the units here are G, expressed as G. The value is expressed as G. And free energy, or G, is the portion of a system's energy that can do work when temperature and pressure are uniform throughout the system, as in a living cell. And you should recall for the exam, you should recall this equation for Gibbs free energy. Delta G equals delta H minus T delta S. And you should know what all these values represent. You should know that delta G represents a change in free energy. And a negative delta G means that it's an exergonic reaction that releases energy. A positive delta G is an endergonic reaction which requires energy and is endergonic. Now, delta H is the change in enthalpy or total energy. You should know that a negative delta H is when heat is released by the system. This is known as an exothermic reaction. A positive delta H is when heat is absorbed by the system, and this is an endothermic reaction. Next, we talked about delta S. Delta S is change in entropy or disorder. If you have a negative delta S, you should know that this is less disorder, while a positive delta S is more disorder and less order. And then lastly, we talked about T value here. T, the units of which are in K or Kelvin. Kelvin is a measurement of temperature, and this is the temperature of the surrounding system, of the surrounding environment in which the system is found. And that Kelvin, you should know, is always a positive value. And we try to set Kelvin to 1 for the purpose of simplifying these equations. Okay? And then you should know when a reaction is always spontaneous. We talked about this in the video. If you're not familiar with this, you can hop to the video, the chapter 8 video for more guidance. But in chapter 8, we discussed how a reaction is always spontaneous or negative delta G when the delta H is negative and the delta S is positive. Another way of saying that, remember, you should know that the other way of saying the same thing is that a reaction is always exergonic or spontaneous when the reaction releases heat and increases disorder. Okay, here's another example. When is a reaction always non-spontaneous? When is a reaction not going to be spontaneous? Guaranteed. And remember, this means that delta G is positive. This occurs when delta H is positive and delta S is negative, right? Another way of saying the same thing is that a non-spontaneous reaction or an endergonic reaction has a positive delta G. This occurs when the reaction is getting cool to the touch because it's absorbing heat, and when it's becoming less disordered. And then do you remember we discussed two situations in which delta G could go either way based on the values of H, T, and S? We talked about these values as, the following. So you do not know necessarily if a reaction is spontaneous if it gives off heat and it decreases disorder. So in this case, it would have a negative delta H and a negative delta S. All right, another situation in which you would not know exactly which way delta G might be going is a reaction where you're absorbing heat and increasing disorder, which means you have a positive delta H and a positive delta S. So recall that from the video. Again, if this is confusing at all, that means you need to go back and watch this part of the video to understand these concepts here. Very important concepts to understanding when reactions are spontaneous and when reactions are not spontaneous. All right, here's some more important information regarding delta G and G values. Remember, the more free energy of a system, the higher the G, the less stable the system with the greater work capacity. The lower the G value, that means less free energy. It's more stable with less work capacity. Remember, equilibrium is when delta G equals 0. This is the point at which the forward and reverse reactions occur at the same rate and describes a state of maximum stability. Remember that exergonic is another term for negative delta G or spontaneous reaction, while endergonic is another term for positive delta G or non-spontaneous reaction. Remember that spontaneous reactions have a higher G value. Here, free energy is G. Spontaneous reactions start with a higher G value, and the products have a lower G value. So the delta G is negative, and these processes release energy. Remember that conversely, endergonic reactions start with a lower G value, low G value reactants, and you need to put energy in. There's a net input of energy with products achieving a higher G value. In this case, the G value is a positive delta G. Remember that in a closed system, it is very easy to achieve equilibrium where no work is being done. And remember that organisms are open thermodynamic systems. Remember that cells need to do 3 types of work: chemical work, transport work, mechanical work. And these types of work require energy, usually in the form of ATP. And through energy coupling, ATP provides the energy required to perform work, to do an exergonic reaction. Remember that energy coupling is when the energy released by an exergonic reaction is used by an endergonic reaction to work, to push it forward. Remember that ATP is adenosine triphosphate, and it is a type of chemical energy which has potential energy in these 3 phosphate groups. When you hydrolyze a phosphate, that releases a massive amount of energy. Remember, we talked about enzymes. Enzymes are usually proteins that are catalysts. A catalyst is a chemical agent that speeds up a reaction without being consumed by the reaction. Remember, enzymes usually end in ACE or ASE, such as sucrase or peroxidase, right? And these enzymes, they have specific substrates such as sucrose, which they bind and they catalyze a reaction. Remember that the way the enzymes work is by lowering what's called activation energy, or E sub A. Remember that without an enzyme present, the activation energy is larger. And in the presence of an enzyme, the activation energy is lowered. And the lower the activation energy, the faster the reaction occurs. Do you remember these terms? Substrate, this is the specific reactant that an enzyme acts on. And then the substrate and enzyme bind together at the active site. There's an induced fit. And this is how an enzyme substrate complex is formed. And then you should also definitely know that enzymes work at a constant rate. Enzymes are not consumed by the reaction they catalyze. Not all enzymes work best at the same temperature. Not all enzymes work best at the same pH value. Enzymes are proteins and they can denature if they stray too far out of their optimal pH or temperature values. Remember cofactors? You should know that cofactors are non-protein enzyme helpers. You should know that organic cofactors are known as coenzymes. These include vitamins. So vitamins are coenzymes, but metals such as zinc is not a coenzyme. Okay, do you remember that? Zinc, iron, copper, these types of things are cofactors, but they are not considered coenzymes because coenzymes are the organic cofactors such as vitamins. Then we talked about inhibitors. Inhibitors bind to enzymes and prevent them from working. Competitive inhibitors resemble the substrate, so they can bind to the active site, preventing the regular substrate from binding to the active site. While non-competitive inhibitors, they bind to another part of the enzyme, not at the active site. Okay, and do you remember how non-competitive inhibitors function? They function by allosteric regulation, which occurs when a regulatory molecule, such as an inhibitor, binds to a protein at one site, like way off away from the active site, and affects the protein's function at another site. Okay, so if I ask you, what's responsible for how non-competitive inhibition works? The answer would be allosteric regulation. Okay. And then lastly, we spoke of feedback inhibition. Remember, this is simply when the product of a metabolic pathway it does serve as the inhibitor for that very pathway. And the reason, you should know the reason for feedback inhibition is twofold. The reason is twofold. One is to conserve initial substrate, and the other is to only produce as much end product as is needed. Awesome. Are you guys ready for our final break time with Gizmo and Wicket, so we can finally get to chapter 9 and be done with the exam 3 review, so we're prepared for the exam. Let's do it. All right, welcome back from break time with Gizmo and Wicket. Let's go ahead and get started with Chapter 9, Cellular Respiration and Fermentation. So one thing you should know for the exam is the equation for cellular respiration. You have glucose and 6 oxygen as reactants, and you should know that the products are 6 CO2, 6 water, and ATP energy. And you should know that eukaryotes net 30 ATP, while prokaryotes net 32 ATP. Next, you should recall the terms oxidation and reduction, because those are very important terms throughout this process. For example, you should realize that glucose becomes oxidized to CO2, and you should realize that oxygen is reduced to water. All right, now you may want to pause the screen at this point and take a screenshot of this, because this is my favorite overview slide of cellular respiration, showing you every single step and the important inputs and outputs of each. So we're going to keep referring back to this slide throughout this review. Remember, let's start at glycolysis. Remember, during glycolysis, these are the things you should know from my class. For glycolysis, we start with glucose. Glycolysis occurs not in the mitochondrion, but outside in the cytosol or cytoplasm. You should know that we begin with glucose as a reactant, and the three products are 2-NADH, 2-pyruvate, and 2-net ATP via substrate level phosphorylation. Remember, the ATP that's formed is formed by substrate level phosphorylation during glycolysis. And then after that, the pyruvate head into the mitochondria. But let's go into glycolysis again in detail. Again, you don't need to know every single one of these steps for my class, but these are the energy investment phases. This is the energy investment phase of glycolysis where 2 ATP are used, and then the sugar is broken in half. That's the glycolysis part of glycolysis, right? Breaking the sugar in half. And then during the payoff phase of glycolysis, remember we made 2-NADH, 4 ATP, and 2-pyruvate. So essentially we netted 2 ATP because we used 2 ATP during the investment phase, and we made back 4 during the payoff phase. So we netted 2 ATP with what process? Substrate level phosphorylation. So again, glycolysis results in 2-NADH, 2-pyruvate, and 2-net ATP via substrate level phosphorylation. Next, let's talk about pyruvate oxidation. Here's pyruvate oxidation. I want you to multiply everything here by 2 because remember, we ended up with 2 3-carbon sugars of pyruvate. We ended up with 2 pyruvates at the end of glycolysis. So everything here is times 2. 2 pyruvate enter the mitochondrion. 2 NADH are formed. 2 CO2 are released. And 2 molecules of acetyl-CoA are formed. 2 acetyl-CoA are formed. So let's go back to our overview and see if this checks out. Looks like it checks out. 2 pyruvate enter the mitochondrion where 2 NADH are formed, 2 acetyl-CoA are formed, and 2 CO2 are formed. Next, the 2 acetyl-CoA we are left with head to the citric acid cycle. So let's go take a look at that. All right, take a look at the citric acid cycle. Again, you have to multiply everything here by 2 because we did not end up with a single acetyl-CoA. We have 2 acetyl-CoAs. So we actually have 2 rounds of the citric acid cycle, which again, remember, citric acid cycle is also known as the Krebs cycle or the TCA cycle, depending on your textbook. Okay, now 2 acetyl-CoA enter the cycle, a total of 1, 2, 3. So 3 times 2, 6 NADH are formed. Here's 1 FADH2, so times 2 would be 2 FADH2 are formed. Here's an ATP that's formed, so let's times that by 2. 2 ATP are formed. And remember, this ATP was also formed by substrate level phosphorylation. And here's 2 CO2s right here, so let's multiply that by 2 as well. 4 CO2 are formed. So again, during the citric acid cycle, 6 total NADH are formed, 2 total FADH2, 2 ATP via substrate level phosphorylation, and 4 CO2 byproducts are formed as well. And the other thing I want you to know for my class, the other thing that I need you to remember is the reason why this is a cycle, and that is because we need to replenish oxaloacetate. See right here, I'm circling. Oxaloacetate is the organic molecule that needs to be replenished, that means reformed, at the end of the citric acid cycle in order to start a new cycle. So let's check our big overview sheet and see if all of that checks out. Yes, it looks like all of that checks out. 2 acetyl-CoA enter the citric acid cycle, so we get 2 rounds of the cycle. During that time, we make 6 NADH, 2 FADH2, 4 CO2, and 2 ATP via what? Substrate level phosphorylation. And what needed to be replenished at the end of the cycle? Remember I said oxaloacetate must be replenished here at the end of the cycle. Looks like it checks out. So now recall that at this point, glucose has finally become totally oxidized. The 6 carbons on glucose have been released. 2 carbons here as CO2 and 4 carbons here as CO2. That's where the C's and the O's went. The carbons and the oxygens of glucose were oxidized. This was released as CO2. And the hydrogens of glucose are now found on these carriers, NADH and FADH2, which are all headed to the final stage to visit the electron transport chain during oxidative phosphorylation, which is the final step of cellular respiration. So let's go and take a look at oxidative phosphorylation in detail. So here is oxidative phosphorylation. This membrane here, recall this membrane represents the inner membrane of the mitochondria, the inner membrane of the mitochondria. That means this fluid out here, this space out here, this is the intermembrane space, the space between the two membranes. And then this space in here, this is the matrix of the mitochondria, recall. Now, in the inner membrane, you have these complexes, 1, 2, 3, and 4. These 4 complexes make up the electron transport chain, while this complex over here, way on the right, this is an enzyme complex, which is also a carrier of protons. This is known as ATP synthase. Now, you should know that NADH, the NADH that were formed, those 10 NADH that were formed during the previous phases of cellular respiration, they're all headed straight here to drop off their electrons. So those electrons zip through the electron transport chain, and the protons are let go of, and those protons can be pumped through these proton pumps. You should know that complex 1, complex 3, complex 4, these are proton pumps. They are active transporters of protons, and they are powered by the electrons that flow. See, when NADH drops off its 2 electrons, those electrons flow through the complexes, powering the complexes to actively pump protons. FADH2, on the other hand, it drops off its electrons here at complex 2. So FADH2 drops off its 2 electrons at complex 2, while NADH drops off its 2 electrons at complex 1. Either way, those 2 electrons go from complex to complex, settling on complex 4. At which point they need to be taken off the ride. Remember, the electrons need to be removed from the complexes. So oxygen, which we breathe in when we respire, oxygen comes along, or 1/2 O2, which is an oxygen, picks up 2 electrons and 2 protons from the matrix to form water, H2O. And you should know that oxygen is serving as the terminal electron acceptor. You should know that oxygen is being reduced to water. You should know that NADH visits complex 1. You should know that NADH2 visits complex 2. You should know that electron flow is what powers the pumps 1, 3, and 4 to pump the protons. You should know that the proton concentration is high in the intermembrane space. You should know that those protons chemiosmose, those protons force their way back in through the carrier ATP synthase. And you should know that that exerts a proton motive force, which provides the energy for ATP synthase to form ATP. You should know that 28 ATP are formed this way, and these ATP are formed by a process not substrate level phosphorylation, but instead these 28 ATP are formed by oxidative phosphorylation. All right, so let's go ahead and check our overview slide and see if everything makes sense. Here we are back at the overview slide, and it looks like everything makes sense. Look, 2 NADH from glycolysis, 2 NADH from pyruvate oxidation, and 6 NADH from citric acid cycle are all on their way to oxidative phosphorylation to visit complex 1 of the electron transport chain to drop off their 2 electrons each. while the 2-FADH2 from citric acid cycle is also on its way to the electron transport chain complex 2 in order to also drop off its 2 electrons each. Those electrons flow through the electron transport chain, and that is a spontaneous process which releases the energy required to pump protons actively with pumps 1, 3, and 4. The proton concentration becomes high in the intermembrane space, and those protons chemiosmose back through ATP synthase, forming 28 ATP. Do you recall 28 ATP are formed via oxidative phosphorylation this time? And do you recall at this point, oxygen is your terminal electron acceptor? Oxygen picks up electrons to become reduced to water. Throughout this process, glucose was oxidized to CO2 while oxygen was reduced to water. And that is your big overview of cellular respiration. All right, next, it is very important to understand the difference between aerobic respiration, anaerobic respiration, and fermentation. None of these three are equal. None of these three are the same thing. So let me explain first the difference between aerobic respiration and anaerobic respiration. For anaerobic respiration, this uses an electron transport chain with a final electron acceptor or terminal electron acceptor other than oxygen. For example, some organisms use sulfate ion, SO4 2-, as the terminal electron acceptor. In this case, hydrogen sulfide is made as a byproduct instead of water. So what does this mean? Let me explain. First of all, again, let me explain the difference between aerobic respiration and anaerobic respiration. Are you ready? So you see here, during aerobic respiration, those electrons are flowing through the electron transport chain, and oxygen is the terminal electron acceptor at the end of the electron transport chain to pick up the electrons to form water. This is known as aerobic respiration. During anaerobic respiration, these are organisms where something other than oxygen serves as the terminal electron acceptor. So remember, like sulfate ion, SO4 2-. It would be the one picking up the electrons to form hydrogen sulfide, H2S. So just so you understand the big picture one more time, do you see everything here, including glycolysis, pyruvate oxidation, which by the way is also known as oxidation of pyruvate, citric acid cycle, and oxidative phosphorylation? If all of these phases are occurring, if all of these things are working, and your terminal electron acceptor is oxygen, that is known as aerobic respiration. However, if all of these things are working, if all of glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation are all functional, but your terminal electron acceptor is a molecule other than oxygen, such as sulfate ion, then that is known as anaerobic respiration. Do you see the difference? The only difference is the identity of the terminal electron acceptor at the end of the electron transport chain. That's it. Now let me ask you this. Do you think that humans are capable of anaerobic respiration? Oh, that's right, Wicket. We are not. We are aerobic respirators. If there isn't oxygen available, we can't start breathing something different, okay? There are certain creatures that breathe oxygen, and then there are other creatures on the planet, usually microbes, that can breathe other substances like sulfate. All right, so in this case, what in the world is fermentation then? Remember, fermentation is not anaerobic respiration. Fermentation is simply when we run low on terminal electron acceptor. So for instance, let's say I hit the gym too hard one day and I'm exercising too hard, and although I'm breathing hard, I do not get enough oxygen in my system. That means I do not have enough terminal electron acceptor. That means I can't pick up all the electrons I want to at the end of the electron transport chain, and as a consequence, oxidative phosphorylation shuts down. the citric acid cycle shuts down, even pyruvate oxidation shuts down. And in the case of fermentation, the only process here of the respiration pathway that keeps going is glycolysis. Only glycolysis continues to function during fermentation. That means that during fermentation, I net only 2 ATP. I also form 2 NADH and 2 pyruvate. All right, let's summarize lactic acid fermentation. Again, this occurs when I don't have enough oxygen as my terminal electron acceptor. Therefore, my body switches to just fermentation, which includes just glycolysis. The products of glycolysis are NADH, 2 NADH, 2 pyruvate, and 2 net ATP via substrate level phosphorylation. However, do you recall what we said during the lecture? That the danger here is running low. You should know the danger is that you might run low on NAD+, because all of your NAD+ is becoming reduced to NADH. And because you have a limited pool of NAD+, this NAD+ pool must be replenished. But the only way to replenish it during fermentation is to reduce pyruvate, is to use NADH to reduce pyruvate. Essentially, you're giving the H from NADH to pyruvate. Thereby, you are reducing pyruvate. You're giving pyruvate H's. You are reducing pyruvate to lactate, which is another way of saying lactic acid. So this is why your body produces lactic acid during fermentation. It's because you need a way of replenishing NAD+, and we do that by directly reducing pyruvate to lactic acid. So again, during fermentation, this gets triggered when there's not enough terminal electron acceptor. The products are 2 ATP, 2 NADH, and lactate. And the thing that needs to be replenished is what? The limiting factor that must be replenished, you should understand, is NAD+. And the way we replenish that is by directly reducing pyruvate to lactic acid. Okay, let's compare that to alcohol fermentation, which is very similar. But in this case, again, there's not enough terminal electron acceptor like oxygen. So the only process that's going on is glycolysis. Again, we are forming 2 ATP, 2 net ATP, 2 NADH. Our glucose becomes pyruvates, right? 2 pyruvates. However, in this step, we are not directly reducing pyruvate to lactic acid. Instead, pyruvate is first changed to acetaldehyde by removal of CO2. Next, NADH can then reduce acetaldehyde to ethanol or drinking alcohol. And that's how we replenish our NAD+. So what do you need to know? You need to know that during fermentation, again, the limiting factor that must be replenished is NAD+. The way it's replenished is not by directly reducing pyruvate, but by first changing pyruvate to acetaldehyde and then reducing that to ethanol. Again, we net 2 ATP, and this occurs in those organisms that undergo fermentation, alcohol fermentation. All right, and I think that was it. I think that those were the important steps. Those were the important concepts and important steps to Chapter 9. I hope that was a good review. Again, I know these are complicated topics, and we didn't have enough time during the review to delve into them, but remember, the full-length lecture videos are all there. Anything that's confusing or you need more input on and more top discussion about, remember, you could go into the full-length videos and watch those where I go into a lot more detail about these concepts. We just didn't have time to delve too deep into them during this review video, but they are there as a resource for you. I know you can do it. Study hard. Study this exam review video. Study your handouts. Go back and watch the full-length lectures. You can do it. Study hard, and I wish you well on exam 3.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.